accreditation body in the world. We've been doing this for over 75 years, and we've been the Australian GLP Compliance Monitoring Authority for over 20 years. Firstly, I'd like to begin by acknowledging the traditional owners of the land on which we meet today, and pay my respects to elders past, present and emerging. In this presentation, I'll be covering the following topics. At various points, there'll be opportunities for questions, and I really encourage you to ask or comment on any you think about. Please use the raised hand function. I just realised I didn't actually start the recording, so I'm going to do it now.
It's okay. It's okay. I have Louise.
Oh, okay. Thank you very much. Sorry. Thanks, Tanya. At various points, there'll be opportunities to ask questions, and it would be great if you would do so. And as I said, if you can just unmute yourself to do that and raise your hand. Tanya or Neil or one of my other NATA colleagues will be monitoring that. If that fails, please feel free to unmute yourself and just grab my attention somehow. I'd prefer not to use the chat function and won't be actively monitoring it. But if you want to put something in there, maybe someone might let me know if there's something of importance in there. I'll also give you my email address at the end of the presentation, and you can email me any questions you might have as well, and I'll try to assist where I can. It's wrong. So let's start with an overview of the GLP principles. This is a graphic that some of you might have seen before. It's one I've been using for some years. When I first saw it early on in my GLP career, I thought it was relevant then, and I think it's still relevant today. It summarises the pathway for any new product, new chemical, new whiz-bang, whatever, from inception to registration and continuing use. So the new product's discovered, it's researched, and if it shows promise, it'll go through preclinical safety testing before it gets pre-approval to move on to the next phase to test to see if it works, which is efficacy. If these trials are successful, it can then go on to be registered and approved for manufacture and sale. These are all regulated activities with various standards, etc., as to how they should be run. And this includes the OECD Principles of Good Laboratory Practice, which only fits into that very small section there for preclinical safety testing. There's also guidelines for GCP and also GMP, which I'm sure many of you in this audience will be aware of those different guidelines and regulations. And depending on the testing or activities that are being done as part of those processes, ISO standards could also apply, and they apply across this whole timeline. It may or may not be required, but it can be used to ensure the technical competence of the laboratory that's performing the testing. This talk, however, will only focus on the OECD principles, not ISO. So the OECD principles are part of the OECD's Environmental Health and Safety Program, whose objectives are shown on this slide, and whose work I'm sure many of you are familiar with. One of the core aspects of this work relates to the Mutual Acceptance or MAD agreement. So what's MAD? When I first saw this acronym, I thought of this guy, Alfred E. Newman. I guess I'm showing my age. There's probably some of you in this audience who may have no idea who that person is. However, I did actually Google MAD and it still exists. They're still writing satirical messages and magazines, but however, it's now on the web. MAD's also not Mutually Assured Destruction. It stands for the Mutual Acceptance of Data Agreement. So that in summary says that if a country adheres to MAD, the regulators or receiving authorities, which are you guys, of that country agree to accept the data from other countries that adhere to MAD and vice versa. They'll take our data and we take theirs. And that is, there should be no repeat testing. The agreement does say that the data must be accepted for review. However, the interpretation of that data rests with you. You may choose to reject the data on scientific grounds or because of a regulatory requirement. MAD is just about reviewing the data. So all OECD member countries that are shown on this map adhere to MAD by default. There are currently 36 OECD member countries, including the US, most of the EU, they're the dark blue ones on the map, Japan, Korea, Australia, New Zealand, Chile, and that all those people's data must be accepted for review. However, as you would all know as well, chemical safety is an international issue, and working with governments outside of the OECD is an important part of the EH&S program. For example, experts from non-member economies often take part in technical work of the OECD, which goes into the development of methods, guidelines, reports, and other products. There are currently 7 non-member adherents to MAD, Argentina, Brazil, India, Malaysia, South Africa, Singapore, and Thailand. These economies are not members of the OECD, but they are full participants to the CBC, or the Chemicals and Biotechnology Committee, for issues that relate to the mutual acceptance of data, or the MAD agreement. And in the working party, they're also in the working party of the National Coordinators of Test Guidelines Program. And as adherents, their data must also be accepted for review. There are currently also several non-member countries who are seeking to be adherents. To become an adherent, there's a process that a country has to go through, and it can take a very long time. Agreements with the OECD are with governments. If a non-OECD member country wishes to become a MAD adherent, the government, not the industry of that country, must liaise with the OECD. It might start with industry lobbying the government, but the agreement has to be with the government and the OECD. They would first become a provisional adherent, which means they must accept data from all MAD adherents, but we don't have to accept their data until they become full adherents. This process can take some time, as a country has to establish or have a compliance monitoring authority, which is appointed by the government. And they also have to establish policies for a GLP compliance monitoring program, including inspections of any domestic facilities that are doing GLP studies. And they must also undergo a successful on-site evaluation visit, which I'll be talking about later. Just before I move on, are there any questions on those topics that I've just gone through? As I said, there'll be plenty of opportunities later on or at the end of this presentation as well. Doesn't look like it, so I'll move on. So let's have a look at the purpose of the OECD principles. The big picture, I think, is to make sure the things we use, that we eat, that we spray on our crops, that are safe. The dog in the middle was mine. He's been dead for some time now, but I like to think that what we do matters, to make sure that everything that we do, our cosmetics, etc., are safe. I also, because I'm old, I like history. And so I'd like to give you a bit of a context about how these principles came about. They began life in the 70s, a period when wide lapels were very fashionable and of strange party food. I'm not quite sure what that thing is. I think it's some sort of pate. They were initially a set of good laboratory practices that came about in response to the quality of scientific safety studies that were being submitted to regulatory authorities for registration or approval of chemicals. There was some fraud that was going on, but there were also some fundamental problems, such as lack of training of staff, lack of planning, procedures not being followed, procedures not standardised, raw data not being recorded and not traceable, poor animal handling, insufficient information on test and reference items, lack of resources, equipment not being checked or maintained, reports not being checked for accuracy or not containing sufficient information, records not being securely stored, etc., etc. They were developed by governments as a regulatory control mechanism to ensure future safety studies would be of acceptable quality and integrity. In 1978, there was an expert group from 17 countries, which included the major players, the US, the UK, Australia was there, WHO, ISO, and they all got together under the OECD Special Program for the Control of Chemicals, and the first edition came out in 1978. In the last decade of the 1900s, fashion became very colourful and food became dainty. It was also when the good laboratory practices established earlier were adopted by the OECD to become the OECD Principles of Good Laboratory Practice. I wish they'd been called Good Studies Practice, because the title would better reflect what they're actually used for. That is the conduct of non-clinical environmental health and safety studies for submission to a government regulatory agency in support of a regulated product. In 1981, the OECD recommended that the principles be used by the OECD member countries. This was also the year of that the decision, as the OECD calls it, concerning MAD was adopted. Decisions are the law, if you like, for want of a better word, in the OECD. And when the decision on MAD was adopted, it said that data generated in the testing of chemicals in an OECD member country in accordance with the OECD test guidelines and the principles must be accepted by other countries for the purposes of assessment and other uses relating to the protection of man and the environment. In 1981, another law, which was this case was called a decision recommendation, came about, which established the need for the GLP Compliance Monitoring Authority. So the first one said, follow the principles. The second one said, we want someone to watch you to make sure that you're following the principles, which is the GLP Compliance Monitoring Authority. And the monitoring authorities were also required to exchange information, which included details of non-compliances and lists of all facilities that were inspected. In 1997, they were revised to what we see today, which is document number 1. In the new millennium, food became healthier and the white t-shirt became accepted as formal attire. The principles also continued to evolve as the world and the industry evolved. However, rather than being revised, which is actually a very difficult process within the OECD series on the principles of good laboratory practice has grown from 11 documents to its current 20, it's actually 25, not 24, to reflect how studies are performed. And this includes the increased use of computerized systems, including the cloud and electronic documents, the conduct of multi-site studies, and incorporating risk assessment. As I said, there's now 25 documents in the series. Most of those are to provide interpretation of the principles to assist both facilities and compliance monitoring authorities. A couple of them, I won't go into detail of all of them, they're available on the OECD website. Some of them cover things like how particular types of studies are done, like there's one on field studies, there's one on in vitro studies, there's one on the general processes that are followed for all studies, such as computerized systems, quality assurance, data integrity, and test items. And there's a few on to cover roles and responsibilities of the various people involved in studies, including study directors, sponsors, and one on multi-site studies, which is very common these days, where studies are done in more than one place, more than one organisation, and of course, more than one country even. There's also ones for how the Compliance Monitoring Authority, that's us, how we're supposed to do our job. And there's even one for you, document number 20, which I'll talk about later. And to date, there are over 40 countries that have adopted the OECD Principles of Good Laboratory Practice. So let's have a quick look at what's in the principles. The scope of the principles is defined at the beginning of the document. And unfortunately, after 20 years, I can actually memorise this. It's a quality system for the organisational processes and conditions under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived, and reported. So as for any quality system, which includes ISO 9000, ISO 17025, or any of those systems that are in place, the principles also make sure that responsibilities are defined, staff are trained and assessed as competent. There are documented procedures. There's some sort of independent oversights, such as audits, and that there are records to show who, what, where, and how things were done. I think it's important to remember the principles do not apply to clinical trials, human or animal, except for some exceptions in the US. The US EPA requires that studies for products that kill rats and insects are done to their GLP regulations. The principle has 10 sections. So this is document number 1, has 10 sections which relate to the points that are put on here. Organisation and personnel, quality assurance, facilities, that's the places where the studies are done, apparatus or equipment, materials and reagents, and that includes computerised systems. Test systems are the thing that is the subject of this, that the test item is being applied to, the rat, the mouse, the crop, or in some cases the analyzer, if it's a physical chemical study. Test and reference items, that is the test item is the article that is being studied. There needs to be standard operating procedures, and there's specific rules on performance of the study and reporting of the study, and then storage and retention of records and materials. Unlike other standards, 17025, 9000, etc., there are some specific things in the principle. And these are there because the principles are a tool for regulators. They're to make sure that regulators get decent quality studies. Some of the areas, the specific ones are things like study director and principal investigator. The study director is the person who is responsible for the study, not the sponsor. And if they can't be responsible for the whole study, maybe because they're not physically in the same place or they don't have the expertise, they then appoint a principal investigator to take responsibility for that part of the study, but they are ultimately responsible. The principles require independent quality assurance. It's nice if they're independent. They must be independent, and if they're not, that is a non-compliance with the principles. One of the things that I've been taught over the years is that QA are the eyes and ears of management. Their role is very important. And also, if we come and inspect and find lots of problems systemically across the organisation, it'll be QA where we'll often have an issue with, because they are supposed to know the principles as well as we do. There are mandatory audits that must occur for every study. Every study plan, every final report, and all the raw data must be audited by QA or audited by QA. There are also specific process-based and study-based audits, and also facility audits that have to be done either for a study and or across a time period. There's specific information of test items. These studies are there to get a new chemical registered. So it's really important that the study, that when the study's done, that the study director and the people doing the study know what they're testing and are assured of what they're putting onto the test system. A whole document number 19, was written 4 years ago to clarify what is needed for test items. The study plan is a unique document that is only listed in the OECD principles. They talk about study protocols in clinical trials, but OECD GLP talks about study plans, and it is the rule book for a study. It overrides SOPs. It's the what must be followed for a study. The principles, though, understand that we live in the real world and things change. So they develop this system of amendments and deviations. Amendments are planned changes, such as a change in study director, maybe a change in the location of somewhere, and they have to be approved by the study director. Deviations are, whoops, a mistake happened, and they just have to be, documented. They have to be documented. The study director has to assess the impact on the study, and they must be listed in the final report. There are specific data requirements. Some of them are common to things like to all quality systems. If you make a change, you have to sign and date the change. GLP also requires that you record the reason for the change, why you changed the data. And with computerised systems, there's a large emphasis on audit trails, making sure that they are readable, they're turned on, and that you can tell what was changed in any computerised system. There are specific report requirements. As I said, this is a tool for you guys. It's a tool for regulators. So in there are things that we hope you guys want to see, and they must be included in a GLP report. And then there's this specific requirement for archiving. Not only do records have to be kept when the study is finalised and at certain time points for records across studies, those records have to be locked away, basically. They have to be locked away under the control of a person who is designated as an archivist. They report to management, and they are the only people who can access the archive. And this is to protect those records. As you, I'm sure, are no doubt aware, you may not see a report that is, you might not see a report for some time after it was done. Could be 5, 10 or more years old. You might want to question what was in that report. And so it's important that that data is kept securely. And that's why the archiving requirements are quite specific. And again, there's a whole document on archiving. So why have GLP? It does promote the quality and validity of the data. It does assure that results are reliable and repeatable, and it does assure that studies are auditable and reconstructible. But there are also limitations. It doesn't assess the science or the experimental design. It doesn't guarantee the validity of the science, and it doesn't guarantee the scientific interpretation of results. Theoretically, someone could do a GLP study that said, I'm going to dose one animal once and I'm going to send that to a regulator. Now, obviously, if we saw that study, I don't think we'd let it be GLP, but you would definitely not. You would definitely say that is not a sufficient study. The study design is the responsibility of the study director, but often they'll be guided by your regulations or your requirements, what you require in those studies. We just make sure that they're done. So why have GLP? For you, we hope that it ensures that data is reproducible and traceable. It helps that the data's there for you if you ever need to look at it again. And it enhances the credibility of data. For facilities, it reduces multiple inspections. It reduces technical barriers to trade because of the MAD agreement. And one of the reasons, which is not anywhere written in the principles, but it's something that I've certainly seen to be true over the 20 years and more of doing this job, any quality system, whether it be GLP, 17025, it probably even 9000, ensures that that the facility operates more effectively and more efficiently. It's one of the pleasures of my job to go to a facility over time. And when you first turn up, they're terrified. The second time, they're feeling a bit more comfortable. And the third time, they often say, gee, I'm so glad I did this. It makes the facility run so much better than it used to. But fundamentally, it's about MAD. People want to be recognised as GLP compliant because they want to take part in the mutual acceptance of data agreement. We've only got 24 facilities in Australia, but when we do our work, we're not working just for you as the Australian regulator, we're also working for all regulators around the world, because what we do here means that the work that is done here can be accepted overseas and vice versa. This slide lists the area of expertise which relates to the types of studies that can be conducted using the principles. As I said before, it doesn't relate to efficacy. And there's also, as part of each of these sections, there's the relevant test guidelines because they're an important part of the MAD agreement. Physical testing is things like the basic routine melting point, boiling point, suspensibility, those sorts of testing that gets done on test items in particular. Toxicity and mutagenicity studies. This is obviously the toxicological properties of a test item and includes such things as the Ames test, etc. These next three, we don't have any facilities in Australia that are accredited to this testing. We did, but we include things like ecotox studies. There's also residue studies is the main component of test facilities in Australia, either doing the field component or the analytical phase, the lab phase. And that's, covers pesticide residues and also animal residues on animals as well, veterinary chemicals. And then there's analytical and clinical chemistry. And this is analytical and clinical chemistry associated with those other types of studies. And this includes such things as hematology, GCMS, biochem, immunoassays, etc. And then, of course, there's always the one you have to have, the other. In Australia, we have a facility that does virus inactivation, which is a non-clinical environmental health and safety study, but it fits under the other criteria. Have we got any questions yet? Done silence? No. Okay. So in this next section, I'd like to have a look at what you and we, NATA, can do to help with the assessment of GLP compliance. Firstly, here, I just thought I'd give you some highlights on how we do assessments, or GLP inspections, as they're called internationally. I wonder, I was going to ask the question, but it's a bit hard virtually. I imagine that some of you have been involved in NATA assessments, either perhaps in other lives, you might have worked in NATA laboratories. Some of you might have had the privilege of observing one of our assessments. But for those of you who aren't familiar with it, I just thought I'd run through the sorts of things that we do at an assessment. I'd also like to emphasise, as I said before, that our program exists to support you as the receiving authority of this data. I do both ISO and GLP assessments for forensic testing, and when I'm doing GLP, I'm always thinking, what does the regulator think? Does the information in the report reflect what happened? Is it transparent? Is it accurate? Is it supported by data? So what we do is we review records. We look at such records as training records, equipment records. We talk to staff. We ask them what they do. We ask them to explain. We ask them where they can find things. We actually go into facilities. We look at archiving facilities. We look at animal houses. We go to field sites. We look at any of the facilities that they may use for GLP studies. And one important aspect is to observe activities. So we're not just looking at records and talking to people. We actually ask them to demonstrate doing work, dosing animals, collecting tissues, spraying crops. One of the best things about my job is the variety of places that I get to go to and things I get to see. And while we're doing that, we're checking are SOPs documented and are they being followed? And that also is the study plan being followed. And we audit studies. That's probably one of the most important aspects of our job, is to audit a study. As the saying goes, the proof of the pudding's in the eating. They need to be able to do studies, and that means we audit those to make sure that the report accurately reflects what the study plan said it was going to do. And if it changed, there are those changes written, documented, clear, transparent, approved, etc. What we look for is traceability. Who did what, with what, when, and how. Suitability. Are the facilities okay? Is the equipment suitable? Has it been validated if it needs to be? And that includes computer system validation, a very important aspect these days of everybody's work. Is it reproducible? Again, can you tell? Would you be able to reproduce who did it, how they did it, and when they did it? Accountability is really important too. Is it clear that the study directors took responsibilities for the study, that management was responsible where they needed to be? If there's a problem with the archives, it's management's responsibility to ensure that's fixed. Did QA do what they were supposed to do? Have they done the audits that they have to do and that they said they would do? And are study personnel recording data correctly, and have they been trained? And once again, security of data is also really important. And that's both physical security, such as for hard copies in rooms, in filing cabinets, etc. But more and more these days, it's security electronically. That's why computerized systems are becoming, the way to do inspections these days. Often the GLP journey, that terrible word, the GLP journey starts with the phone call, I need GLP. And because I'm the GLP Program Adviser, I tend to be the one who gets those calls. But most of the time they don't need OECD GLP because the scope is, as I said, so narrow. Non-clinical environmental health and safety studies for regulatory purposes. They need ISO GLP, good laboratory practice. They're not doing environmental health and safety studies, but they're probably doing routine testing for clients, QC testing, clinical trials, that sort of work. So if they are doing real GLP, then we'll provide them within the information that's available on our website. You guys can access that too if you wish. And if they're still keen, we then conduct an advisory visit. So we can get an idea of what they do and where they do it, and are they ready to apply. After the advisory visit, they get a letter. It's not a report, but it gives them an indication of what they need to consider before applying. And when they're ready, they can complete an application form and pay an application fee. The advisory visit is also an opportunity for them to ask us questions and for us to ask them questions, but it's important that we don't consult. We're not a consultation service. And we also require that they do at least one study they consider to be compliant before they can have an initial assessment. So they need to plan and document the system, check it works by doing the study, and then report and archive the study to be able to demonstrate they comply. Archiving is a critical part of a GLP study or a GLP facility, and so that's why we require them to go through the whole process. We also need to witness an activity, as I mentioned before, to see whether they do follow their SOPs and they've got the competence to do what they say they're doing, dosing animals, necropsy, etc. So it can take one or more visits before an assessment is complete. It's fairly rare that we can see all the aspects that we need to see in one visit. After the visit, they receive a report which details any non-conformances that need to be addressed. They then provide documentary evidence to address these, and once they're satisfactorily been addressed, we then recommend that they become part of the Australian GLP Compliance Monitoring Program. NADA is a not-for-profit members association, and we charge them for what we do, fee for service basically, until they're recognised, which includes time, expenses, etc. Once they're recognised, they pay an annual fee, which it will cover approximately 3 days on site and probably basically one study type. If they do lots of studies or they've got lots of study directors, we charge an additional effort because we have to spend more time on site. Once a facility is recognised, they become part of the Australian GLP Compliance Monitoring Program, and they're listed on our website. After 1 year, we go back and do a study audit if they've done one, just to make sure that the system is properly in place. If not, if they haven't done a study, we waive that and we don't go back until 2 years after the first visit. That's called their first reassessment, and it's the same pretty much as the initial assessment. We look at all the processes, review all their SOPs, we observe activities, and if all is going okay, they then go on to a routine assessment cycle, which is a visit every 2 years. After 2 or 3 visits, I have to say the focus of those visits tend to err towards study audits, particularly if there's been no changes. If the facility is very stable, they haven't introduced any new systems, then the most important thing is to get study audits done. We sometimes need to do follow-up assessments because we need to ensure that the conditions have been addressed, the documentary evidence isn't sufficient, and depending on what happens, we may also impose a shorter reassessment cycle if we think we need to go back sooner. We also do what's called for cause inspections, which are done at the request of a regulator. We actually have a for cause inspection in the pipeline. Well, we have to do one for the FDA has requested that we do a for cause inspection of one of our GLP facilities. And if ever you have a study that you would like us to audit, please feel free to contact me. This includes overseas studies. That's how the FDA one came about. If you have a study done by overseas that you have questions about, please contact me and we can see what can be done about it. We can't inspect a facility just because they want us to. We often get requests from overseas, China in particular, for us to come and call them GLP compliant. For Australian facilities, they have to be doing GLP studies. We can't just inspect anyone doing any work. They have to be doing non-clinical environmental health and safety studies for regulatory purposes. And we also don't inspect facilities outside of Australia. These facilities should be inspected by their own national GLP compliance monitoring authority. Or if they're from a country that doesn't adhere to MAD, like China, we also don't inspect. We only inspect at your request. If you want us to go to China, we'll go. There are some countries that do inspect non-domestic facilities, but this is because they've been asked to by their regulator to do so. And it's important that you realise that even though these inspections have occurred, MAD doesn't apply to that data. You're under no obligation to accept it. On occasions, we are able to invite you to observe us doing GLP assessments, and we've actually managed to do that this year. And I think hopefully 2 of you at least will get to come from the APVMA. We'll be able to come along and observe us doing an inspection. So the next part is what you can do to ensure GLP compliance. As I mentioned, there's an OECD document to assist you in what to look for. It's called document number 20, Guidance for Receiving Authorities on the Review of the GLP Status of Non-Clinical Safety Studies. And it's available to assist you. Some of the points in this document include the fact that the full name and address of a test facility, test sites, sponsors, study director, and if applicable, principal investigators, should be verified against the annual overview or the NATA website. I'll talk about the annual overview shortly. And these documents also allow you to check the compliance status of these facilities and test sites. There must be full identification and characterisation of test items and reference items, including expiry date and information on purity and composition, as well as stability, concentration and homogeneity when these test items might have been mixed into a vehicle, water, adjuvant, whatever. Where this information comes from must be clearly indicated in the report. And if it's not, if you can't tell where it came from, then there could potentially be a problem. This information might have come from a certificate of analysis. It could be based on information provided by the sponsor. It might even be a label from a pre-existing product. But if the study director is unable to verify the quality of the information of the test item data, then the validity of the reported data might also be questioned. And you may question the claim of GLP compliance and or consider rejecting the data. Experimental start and completion dates are also a necessary requirement of a GLP report, and this includes the study completion date, which is the date the study report is signed. And this can be used to verify the compliance status of the test facility and, if applicable, test sites at the time the study was carried out. They may no longer be part of a program, or they may have not actually entered a program at the time they send you a report. The final study report must be signed and dated by the study director. It's called the study director's statement. And in that statement, there will be a GLP compliance statement, which indicates the extent of GLP compliance. If it's a multi-site study, that study director's statement can either include or refer to claims from principal investigators. For some multi-site studies, the PI will issue a separate report with a separate statement. For others, they might just issue data with a statement. But regardless, it should be clear that the PI is making a statement for the work for which they're responsible. And it's important you realise we don't look at every single study, and therefore you need to verify the status of the facility and test sites, because, as I said, we don't look at every study. We look at a proportion. You may have seen in some reports that sometimes they exclude parts of the study from the GLP compliance statement, like weather data, clinical pathology results. The impact of any non-compliant phase on the study must be indicated. It might say it wasn't done under GLP, but it was done under 17025, for example. It's your call if you think that exclusion of a part of a study as being non-compliant jeopardises the status of the study as a whole. Again, you might consider rejecting the data if they've claimed most of the work as not being GLP. The other statement that must be in a report is the QA statement. This lists the types of studies and the dates of inspections. This is the QA audits and the dates of reporting those results to the various people, test facility management, study director, etc. What you're looking for in those statements is that it demonstrates that there was adequate coverage by QA of the plan, the study conduct, and the report. And it must also confirm that the report reflects the raw data. If this information is incomplete, the GLP compliance status of the study might be in jeopardy, and you might also, again, consider rejecting the study. There also needs to be a description of the methods and materials, including reference to test guidelines where they're applicable. And this information should be sufficient to verify the adequacy and the correctness of the methods and materials used. If it's incomplete, then again, you might wish to have to think about whether that is a GLP compliance study. An incomplete summary or a full description of all results, including calculations and statistical evaluation, would also jeopardise the GLP status of the study. This might include inconsistent results, unexpected variability, or data that seem to be not in line with other scientific sources. And this may raise concerns about the GLP compliance of a study. If it does, feel free to contact me. There are some other aspects that you should be looking for. Deviations, as I mentioned before, are mistakes that happen that aren't planned. They need to be identified as such in the study report, and the impact on the validity of the data needs to be discussed with the study director. While deviations are not always indicative of poorly planned or executed studies, they might raise concerns about the GLP compliance of the status of the study, depending on what they are. It is one thing that we make sure that those deviations are included in reports so that you can make an assessment of whether you think they matter or not. Obviously, unrealistic explanations or interpretation of the data in the evaluation and discussion of the results and or conclusions might also raise some concerns. Once again, as mentioned before, archiving is a really important aspect of this, and the storage location of all study-related documents and materials, including the study plan, samples of test and reference items, specimens, raw data, and study reports needs to be included in the report. And for multi-site studies, it should be clear which records are kept by test sites and which records are kept by the test facility. Lastly, sometimes reports need to be amended, and if there are any corrections or additions to report, these have to be written as report amendments. Sometimes the report is amended, like the complete report is amended as an amended report, but regardless, it's got to be clear what's changed. The reason for the corrections and or additions need to be explained and must be signed by the study director. Any report amendment or amended report has to also have a revised QA statement because QA must audit that amended report. If any of those are missing, then you can perhaps question the GLP validity. Any questions on that bit? Okay. So, as mentioned in the last slide, one simple way that you can check whether a facility is in our program or not is to go to our website. And you can also check to see if the study has been inspected by us. The way to do this is you go to the NATA website, www.nata.com.au, and select this button here, Find an Organisation. You'll get this screen, and then you can filter. You can select various filters here. This one I've selected the accreditation or site number, but you can select name and put in either the name or the number and then hit the search button. Once you have done that, you'll possibly get more than one option, but hopefully you'll be able to know which option you want. When you click on that site, it'll take you to the scope, and the scope of recognition is not like the scope of accreditation for a 17025 laboratory. The scope of recognition for GLP facilities is this little tiny bit here, this bit, which is non-clinical environmental health and safety is conducted within Australia for regulatory purposes. This document also includes what we call the types of studies, which is this bit down the bottom, which we talked about before. So this is the types of studies that we mentioned before: analytical and clinical chemistry associated with non-clinical studies. This is the products that these people can work with, and these are the determinants that we have, the types of studies that we looked at. But please be aware that if a type of study or a survey, if this doesn't list the type of study that you're looking at, it doesn't mean it isn't compliant. It just means that they haven't been inspected for that type of study. If they're in our program, they can claim compliance for any study that falls within the scope, non-clinical environmental health and safety studies, blah, blah, blah. When we go there next time, one of the requirements of the principles is that all GLP studies must be listed on a document that's called the master list. And that master list is sent to us prior to a visit, which we review in depth. We look for any new study types that we haven't inspected before, and we will make sure that those new study types are inspected. And if they're compliant, it can be added to the types of studies. So, as I said, it's different to 17025. 17025, a laboratory is accredited for certain tests, and that is their scope. For GLP, the scope is broad. And we don't look at every study. If a facility is compliant, they are compliant to do any GLP study. We sample across the types of studies that they do. Firstly, I just wanted to introduce some concepts about the Working Party on GLP that you may not be aware of. The OECD Working Party on GLP is one of the subgroups under the Chemicals and Biotechnology Committee, the CBC. It meets annually to discuss GLP issues. It often includes a whole day spent discussing technical issues to improve harmonisation. This day often leads to new documents or revisions to a page on the OECD website called the Frequently Asked Questions page. This page is accessible by everybody, including yourselves, the public. And an example of this was as a result of one of the issues that we discussed in 2022, there was clarification about the requirements for homogeneity, concentration and stability of test items that were prepared for field trials. And the frequently asked question page was updated to reflect this. The Working Party also develops new documents and develops and conducts training courses for GLP inspectors. These documents and the training courses are really important because predominantly what we do is about exchanging data. And therefore, I'm talking about both data on compliance facilities and also you guys getting good quality data. And therefore, we strive for consistency between countries. And the latest document was published just last week, and I think I advised people yesterday of this document. It's the first of its kind. It's called Supplement Number 1, GLP and Cloud Computing Supplement to Document Number 17. So that document came about after various discussions at Working Party meetings, etc. One of our other roles is to assist countries to develop and implement GLP compliance monitoring programs in their countries. We want people to be part of the MAD agreement because we believe it leads to better health and safety outcomes. A very large chunk of the meeting is spent discussing reports from on-site evaluation visits, especially as we're playing catch-up from COVID. These on-site evaluation visits provide a mechanism to increase confidence in the compliance monitoring practices of OECD member countries or non-member countries. Such confidence helps monitoring programs to obtain and provide timely and accurate information about test facilities to their respective receiving authorities for use in evaluating the quality of data submitted for their review and assessment. At the recent meeting this year, 7 OE on-site evaluation reports were discussed at this meeting, and all of them only had minor deviations or recommendations listed in the report. These compliance monitoring authorities were found to be implementing the two documents that are part of the OECD series, which is document number 2 and document number 3. Document number 2 is about how the compliance monitoring program is set up and established, and document number 3 is about how we do inspections. Just so you know too, I'm not able to give you those reports. Only the evaluated monitoring program can make those reports available to people. Our on-site evaluation visit is in August, and I'll be talking about that briefly at the end of this meeting, this topic. I've just completed a 3-year term as chair of the Working Party, and with over 42 countries and with their own domestic pressures and strains, it can sometimes be seen like herding cats. Now, I know this is a cat herding cows, but it just made me laugh. That's why I put it in. Luckily though, we work on consensus. We don't vote. If an idea is being discussed to reach a conclusion, we need all members to agree. If one person who sits around that table objects and we can't find a way to agree on what's proposed, then the idea doesn't get up. This can mean there can be a lot of discussion, but in my experience, I've also had to learn to listen. If I want to change or convince someone, I need to understand where they are coming from. It also means that once we've agreed on something, we're obliged to support it. I think one of my roles as chair was to encourage full participation by all members, which can be challenging for some whose first language is not English. The catch cry at the meeting is silence means agreement. It was a pleasure to be the chair for the last 3 years, and even more of a pleasure to work with the members of the Working Party for over 20 years. One of our strengths as a Working Party, I think, is that the people who sit around the table. With regards to Australia, we currently have, I think it's 22 facilities. Predominantly, these are involved in crop studies, but they also cover toxicology, veterinary studies, analytical chemistry, clinical pathology and histopathology, and I talked about the facility that does vaccine studies. Just a reminder that NATA's program only covers Australian facilities. We can't inspect New Zealand test sites, and MAD would only apply to those sites if they've been inspected by IANZ, for example. We're often asked, can't the New Zealand test site come under us? The answer is no. I should have actually changed this to the latest document, but these two are also very new documents. We've been inspecting against these documents now for approximately 12 months, I think. Document number 22 and 23, they're the latest documents to be released. Like all documents, they were drafted by members of the Working Party, and then they were also sent out for public comment by industry regulators and facilities before they were published. They are called advisory documents, but it's generally expected that the facilities will implement and follow what's in them because they're best practice. Document 22 promotes a risk-based approach and emphasises the Alcoa Plus principles. I'm not sure how many of you are aware of Alcoa Plus, but that's attributable, legible, contemporaneous, original, and accurate. And the plus refers to complete, consistent, enduring, and available. Basically, can the data be trusted? There's a focus on electronic data in this document, but it applies to all formats of data and records. As detailed in the definition in document 22, assuring data integrity requires appropriate quality and risk management systems, including adherence to sound scientific principles, good documentation practices, and training of personnel. We've only done a few assessments against these documents, and many facilities already comply with most of what's in there. If they haven't already, we require them to do a gap analysis to identify where improvements in their processes are needed. One of the issues we are seeing that's new, if you like, in the document is there is a requirement to control blank templates or pro formas that are used for data capture to reduce the risk of recreating data. Document 23, this document actually replaces a previous document, document number 4 on quality assurance. Again, most facilities already comply with what's in there, but one of the biggest changes is a concept of risk-based assessments. Often they've been making the decisions on what to audit based on risk, but they haven't formalised it or documented it. That now has to occur. There's another companion paper, I suppose, to document 23, which is called, which is a position paper on quality improvement tools. It talks about things like corrective actions, those sorts of things, which aren't a requirement of the principles, but obviously are really important if you're going to be doing internal audits. This last slide has my details on there. louise.calder@nata.com.au is my email. That's my mobile number. I'm more than happy to get any emails from any of you anytime. And because as I said, we work for you. And that's why we like to do these presentations as well. I hope you did find this useful, and thank you very much for attending, and please, contact me anytime. Thank you.